Okay, so we're moving on to directly to our first panel session. We have two panel sessions, one focused on the need coming from the drug discovery industry, and the second one focusing on how AI can then come in to help. So the first panel session is on the drug discovery bottlenecks and how AI can, help address these bottlenecks. And it's going to be moderated by Dr. Diane Joseph-McCarthy. So I would like to welcome Dr. Joseph-McCarthy and also all the panelists up on stage, please.
So I'll give all the panelists a moment to get up on the stage. And I think while they're doing that, I'll just get started. So I'm Diane Joseph-McCarthy. I'm the Executive Director of the Bioengineering, Technology, and Entrepreneurship Center, or BTEC at BU. I'm also a faculty member in biomedical engineering. And perfect.
We're going to try our best.
There, yeah. So before coming to BU in 2019, I spent most of my career in the pharmaceutical and biotech industry. And so I am really delighted to be moderating this panel on drug discovery bottlenecks and how AI can help. And the panelists who've all just assembled are all world-leading experts on drug discovery and the use of AI. So I'm going to quickly introduce each of the panelists, and then I'm going to turn it over to them to say a little bit more about their backgrounds. So we have Etai Jacob, Senior Director and wave when I say your name, Head of Data Science and AI Early Discovery, AstraZeneca. We also have Ashwini Ghogare, Head of AI and Automation in Drug Discovery, MilliporeSigma. Juli Klemm, Program Director, Center for Strategic Scientific Initiatives, National Cancer Institute. Pat Walters, Chief Data Officer, Relay Therapeutics, and Rob Moccia, Senior Director, Machine Learning Research Lead, Pfizer. Okay, so I'm going to ask the panel several questions, and then assuming that there's time, I'll open it up to the audience, so keep that in mind and be thinking about questions that you want to ask. But the first question is that I'd like each of you to spend 1 or 2 minutes describing your role and the areas in drug discovery in which the teams you oversee or interact with are most involved. And Pat, why don't you start?
Okay. So I lead a group that's involved in computational chemistry, but there is a large machine learning component to that. So we're using machine learning all the way from the very beginning of drug discovery projects using protein structure prediction to develop hypotheses. Protein structure prediction has now become an integral part of structural biology in industry. We're using machine learning as part of hit identification efforts in searching through large multi-billion molecule databases throughout a lead optimization process. We're developing machine learning models that are being used for both on and off-target prediction. So really, machine learning is spanning our work all the way from investigating targets all the way through developing a clinical candidate.
Yeah, we can just go down the line for this one.
Perfect. Thank you. It's great to be on the panel here in such a, I would say, amazing space and good energy here. I'm Ashwini Ghogare. I lead AI and automation for drug discovery at MilliporeSigma, EMD Serono. I think all of you know us in different names and in different parts of the world. Essentially, what my team is focused on is developing ML machine learning models for new molecule drug designs, but focused on small molecules to start with. We focus on how do you essentially integrate both on designing molecules for drug discovery early stage, as well as then converting into retrosynthesis pathways, and then connecting that to automation. And for us, one of the biggest problems that we face and that we're trying to solve is, yes, the great generative models, right? They can design all kinds of molecules, but how synthesizable are these molecules? And do they meet the ADME criteria that's important for drug design? And that usually is the bottleneck in all our medchem projects. And so we're trying to tackle this problem right from the get-go of introducing synthesis accessibility as well as ADME when you do design. And then bringing these predictions into automation where we make these compounds, test them, and then bring that data back into the machine learning models to create the loop, or the lab in the loop, so-called. So super excited to talk about it. And thank you.
Hi, thank you very much. I'm Juli Klemm from the National Cancer Institute, and my colleague Sean Mooney gave a fantastic overview of the National Institutes of Health, where NCI is one of those 27 institutes. The NCI is the nation's leader in funding cancer research and supporting our National Cancer Plan. And NCI provides sustained support for basic research into all aspects of cancer. And so an important role NCI and the NIH more broadly plays is supporting that sustained basic research into disease. In terms of some of the activities within NCI, specifically supporting AI, I coordinate the NCI's Informatics Technology for Cancer Research program, which is an investigator-initiated grant program for supporting informatics technologies across the development lifecycle for all types of cancer research. And increasingly, the projects coming in through this program are very much focused on leveraging AI approaches to advance informatics technologies for cancer research. In addition, I co-chair an NCI Artificial Intelligence Working Group to coordinate the NCI's efforts across our very large institution around how NCI is advancing and supporting AI for cancer research. And that also allows us to provide centralized place for engagement with external organizations who partner with NCI toward these common goals. Thank you.
Okay, great event. Thanks for inviting me. So I'm Etai Jacob. I'm leading the AI research group in early oncology. That means from early discovery till phase 2. So we do, in my group, we do target prioritization and identification, and then a lot of work around translational medicine in the cancer space. So the group is organized in a methodological way. We have knowledge graphs, we have mathematical modeling, we have AI, deep learning, computational statistics, and algorithm development, and on top of doing understanding better the biology and trying to build models that enable us to bring together knowledge from different domains into understanding the biology. We do a lot of work around the translational space where we try to understand better the patient population and how they would be aligned to the different drugs. This is critical, and I think it's very encouraging to see how the NIH strategy is aligned with really getting data in all different domains, including the clinical real-world data. And really, what we try to do is to link as much as early as we can in the early discovery, really the patient population, understanding the human toxicity, and where we could really improve our ability to find drugs that are successful.
Hi, I'm Rob Moccia. I'm from Pfizer. I lead a team in our machine learning research group, and our task is actually focused entirely on computational biology. So none of the chemistry stuff, I know nothing about it. I'm going to pass on all those questions, just so you know. So yeah, we focus on computational biology, and at the moment that really means largely omics, but I can even go deeper than that. Within omics, the most common use case right now, at least in our company, is really around single cell and single nucleus, various methods around that. And we spend a lot of time trying to find models that we think do useful things for drug discovery. At the moment, that actually means a lot of deep interrogation of what these models are able to learn, what it seems that they're not really learning, in finding the places where we can actually recommend to our computational biologists that we have a tool that's going to give useful insights for specific tasks within drug discovery and steer them away from things that may actually be splashy but give misleading insights that aren't actually going to help us find the right targets or find the right chemicals that go after those targets.
Okay, I'm not going to try to take this out of here. So the next question, which you touched on a little bit, Rob, is from your perspectives, which are all a little bit different, where in the drug discovery process is AI currently having the greatest impact? And why don't we start with you, Rob, so we'll go in a different order.
Sure, yeah, I bought that one. Okay, so this is the part where there's the bottlenecks question or the part where it's working?
Where is it working?
Okay.
Where is it having?
Yeah, I think actually the previous speakers did a really good job of setting the stage for this. You already heard a lot about the protein language models. For me, that's the easy go-to answer in the computational biology space. I think protein language models already out of the box are very useful for different tasks. That's actually why my group has not spent a lot of time directly on the protein language models, is because they're working and there's not much for us to do. They're already in the hands of computational biologists and being used to make very useful insights. We can use those, though, to build when we get to the part about where things aren't working. Maybe there are ways we can leverage the parts that are working to shore up the parts that do not.
So I'm definitely aligned with what you're saying. So I think in general we have small molecule, biologics, and computational pathology. I think these areas are covered relatively well in the space with AI. I think when you think about these three domains have a lot of data organized in a good way that enables really to build the models. We are less focused. My group is less focused there. We are more focused on integrating those modalities together and trying to link it to the biology and the clinical MOAs and work there.
Thank you. So, not running a drug development program ourselves, but what I think we can see is that, as was recognized with this year's Nobel Prize, the progress in those structural approaches has been, fundamentally advanced with the tools such as AlphaFold and David Baker's Rosetta and so forth. So I think, there's, very clear revolutionary advances there. But as we've heard in the talks this morning, especially the first talk, some of these approaches to using pathway approaches to identify targets is starting to be advanced with AI approaches, and specifically thinking about traditionally undruggable targets. How can we use, synthetic biology approaches, use approaches that to look at the pathway, to look at where there have been targets traditionally not druggable, how can we then find other targets in the pathway that actually can be druggable? And so I think those are the places where I think there's some exciting developments happening.
Yeah, so I think really great points raised, sort of expressed by the other panelists. I think where we'll add a bit more color to this, I think models in general for small molecules especially are quite democratized at this point. I think there are multiple models, including the reinvent model that AstraZeneca released that all of us mostly use, or they could be transfer models, transfer learning models, the GAN models, and many, many different kinds of models that I think they are available and quite democratized. But I think the challenge that still lies is how do you, again, I bring this whole point, which I'm extremely passionate about, is how do you tie this to synthesizability? How can you make these compounds? And that is a really difficult challenge to solve. I know there's a lot of research happening in various research groups on tying this back, whether it's ML-based or retrosynthesis-based, but really tuning your models to, or each of your multimodal models look into synthesis is super important. And there's a lot of research happening in that area, but I feel like there's more that can be done.
All right, I'm going to disagree with a few of the other speakers. I think, first of all, protein structure prediction is great. It's providing insights. I think from a compound design standpoint, and I'm talking more from the standpoint of chemistry, I think in a lot of cases, AlphaFold structures are really not ready for prime time. If you take a look at the literature and you take a look at what people have been doing trying to dock small molecules into AlphaFold structures, there's really only one case I'm aware of that's been successful and several that weren't successful. I think I agree that some of these generative models are democratized, but they also still have a nasty habit of creating molecules that are just silly and not chemically stable. And I think we need better ways of approaching that. You know, one of the places that I think we've really had a lot of success with machine learning is in applying active learning. So in applying active learning, we can develop machine learning models that are surrogates for more expensive computational methods, and we can do things at a larger scale, and there's a lot of work going on right now integrating physics-based methods with machine learning, which I think are, again, letting us do things at a much larger scale.
Okay, well, we touched upon this a little bit. The next question was supposed to be, what are the key bottlenecks and gaps in drug discovery, and how can AI help? So I think we're going to mix it up a little bit. So maybe I'm going to go to Ashwini first on this one, if you'd like to elaborate. And then people can jump into, we don't have to just go in order. So if there's something that you want to say, please do.
Yeah, and I think, Pat, probably you agreed with us by disagreeing that there are these Gen AI models that aren't perfect, right? And I think we all agree on that. One of the fundamental, I think, gaps in any kind of model, especially in small molecule, is the lack of the positive and negative data, right? All of us are working with positive published data, everywhere in the world, but there's really lack of negative data that we don't publish. And if we publish, we don't trust that data. And so how do we expect our models to have better prediction if there is no negative data to train them? And this is a fundamental problem. I think we talk about in multiple forums, but it hasn't been addressed. And one of the solutions to this is really doing experiments. I don't think we can get around this problem. There is no synthetic data, no matter how you work on this. Chemistry and ADME is such a complex problem. The more we can actually generate some of this data and on a focused target, right? I think we'll get better quality of both positive and negative data, and the models will get better. And we've been trying to accomplish this exactly in the labs that we've developed to try to tackle this problem. It's going to take time. It's going to take money, a lot of funding to get that state. And I think some of us also talked about collaborating on some of these efforts to build a lab that could allow all of us to share that data, generate that data, and share as well. But I think that's a fundamental problem in all of these models that may be democratized, but for them to be used successfully.
I agree. So maybe Etai, would you like to go next?
Sure. So I don't think, Pat, that we are in a disagreement. I think that the question was like where AI has made the largest impact. So I think that in the biologics, it made more impact than in biomarker discovery, right? So I think, but I think you made very good points, and that's a good emphasis. Now I think, when you think about data in biology, right? We have different levels. We have the data at the molecular level, at the cellular level, at the tissue level, at the system level, patient level, and population, right? So we have population level from clinical trials, we have real-world data, and then one of the things that we really need to have more is system level, really, so connecting, the dots between different tissues, right? Between to understand better how drugs works to replace animal models that are not really reflective of human biology with something that works better. So I think generating more data that will enable us to bring together these different system level components will help us a lot. This will maybe give us more opportunities to find biomarkers that will enable us to improve greatly the success of clinical trials that is now around 1 out of 10. for NME that enters the clinical study, right? So 1 out of 10 for launch. It doesn't mean that it's going to be a drug that's going to work. Work, but not maybe successful and disseminated in the community, but still. So that's very low, and I think that's where we need to generate more data, so in a few years we'll be able to come up with biomarkers in a timely manner.
Go ahead. Yeah.
So cannot disagree with data. Of course, data is a huge problem. I'll add to that too metadata. So a lot of times, even after we collect the data, it might not actually have contained all the metadata we need to control for various covariates that we know are influencing models. Also harmonizing across the data sets. So for things where we're doing cell type annotation, even being able to test how well your model's doing, a lot of these data sets have curated labels that will vary based off of the author. Someone might do T_cell, someone might do T cell, someone might be CD4 T cell. Also various levels of granularity. Most of the biologists I've spoken to, they're not as excited about our ability to discriminate T cells from B cells. They're more interested in our ability to look at various types of fibroblasts and what they're actually doing. Or for instance, macrophages, are these pro-inflammatory or are these more in a healthy resting state? So getting more at finer levels of granularity, and that will require the data actually being labeled that way for us to really make sense of it. And then the one other part I'd add to it on the data is that we are very human-centric for obvious reasons. And I actually think, at least in the field of single cell that we've worked most on, it's held us back for a couple of reasons. One is that we're not seeing diversity of biology across evolution that I think actually could inform the models. So if we were training over multiple species, I actually agree with others who have suggested the models might actually learn more generalizable patterns. But also within drug discovery, we don't actually do the experiments on humans until it's safe to do so very late in the process. So we're using model systems, and our model systems don't always reflect the human biology. So if we could get to a place where we had more data on these model systems and started to learn, for example, shared latent spaces that help understand the similarities and differences across them, that would assist in model choosing for our projects, what model is going to be the best model system for different aspects of it, but also the translatability. When our chemists are doing a great job of getting molecules to do what we're asking them to do in these model systems, and then they fail later because the model system actually wasn't capturing what it needed to do in humans to impact disease.
Juli?
I feel like every time we come to a meeting about use of AI, it always comes down to data, and that's of course not surprising because that's what drives these models, and so that I think that I'm not surprised that we've come to that in this conversation, and I think it raises really interesting opportunities based on the goals of this meeting. Are what are opportunities for open innovation? Where can we share data and collaborate on data, but then compete on how that data is used. So I think that's exciting ideas we can also explore in the breakout. So I think I'll just agree with other points that have been made here. Thanks.
Great. Pat?
Yeah, so I'll continue on the theme of data, and I think one of the things that's holding the field back, especially in terms of ADME prediction, is just a paucity of good publicly available data. If you take a look, all the publications that are out there are really based on 2 data sets, so MoleculeNet and the Therapeutic Data Commons. Both of these data sets are frankly terrible. They contain numerous mistakes. They've got data sets that are curated from dozens of papers where the experiments weren't run in a consistent fashion. So one of the things I'm really excited about is an effort that was just funded by ARPA-H. There's an open ADMET effort that's being chaired by Jamie Fraser at UCSF and John Cadera at Memorial Sloan Kettering. And the aims are twofold. One is to generate large, high-quality ADME data sets and make them publicly available. The second, which I'm even more excited about, is to do structural biology on off-targets, because for the most part, structural biologists have completely ignored things like the hERG channel, cytochrome P450s, PXR. So I think if we can start getting structures of ligands bound to these off targets, combine that with these high quality data sets, we can really get some insights that will push the field forward.
Yeah, I couldn't agree more. And I think that really leads into this next question, which is on, why is open innovation important and how can it accelerate drug discovery in the future? We can start with you, Rob.
Sure, yep. Yeah, so I think it goes back to the data in part, right? So we need more of it, and if data is becoming siloed, number one, it's impossible to compare models if you don't have access to the data that models were trained and tested on. But also, I see it as a collaborative effort, right? We need to accumulate everything that we're learning, share it, almost the way we in industry call like a pre-competitive type thing. This is not what's going to give a company a unique advantage. If we all had access to more data, we could then have the competition later when it gets into the clinical space. But it's really about making everything available so everyone has the same stuff to try to improve upon these models. I think, to me, that's the biggest part of it, to avoid silos, to avoid restrictive licensing of tools that lead us to not be able to utilize them for basic tasks, things like that.
Right, and to be able to compare the models, that was a good point as well. But.
Yeah, and I think the example we had earlier, CASP, right? So let's imagine a world where we could have CASP for any problem that we're interested in, right? One of the issues is that CASP was the problem is well defined, right? But when you think about single cell, right? And then, so how would you define, what's granularity are you going to use to define cell types, right? You would think one thing, immunologists would think one thing, and then somebody else would think something else. So I think that also defining the problems in a way that you could benchmark or build a metric, right? That's something that is required, and I think that this, sharing data and models and having an organizations that oversees that, I think definitely open source. That's something that will help us to maybe define the problems better and build a metric that is not based on peer-reviewed of one specific data set that is not the best. So I think that's very important.
Yeah, I think this point about having common benchmarks against which we can measure progress is really exciting and important. I think CASP is obviously a great example, but I think there are other examples in the AI field where benchmarks really help move the field forward because we could all understand where progress is being made and what we were being measured against. I think open innovation is also about just letting the scientific process play out and understanding what has been known in these very complex disease areas is very important for building on that knowledge base and continuing to have a shared understanding of the state of the science so that progress can be made. So I think open innovation is a naturally part of the scientific process that I think can raise all boats.
I know we didn't want to go in specific order, but since we are, I will say that I think it is, we do have to catch up on the ADME initiative that I'm super interested in. But I think it's an important point that all the panelists have raised here is that before we start sharing data, we have to share how we do experiments. Because if all of us do ADME experiments differently, we cannot share that data. So we're going to go back to the same problem that we started with, is everybody having different data sets, publicly known data sets, but we cannot use because we all run these experiments fundamentally differently. And we cannot, as a result, compare these data points. So I think we do have to recognize that it's important. So these collaborations, and I think and that's where open source and innovation is important, because if there's a consortia where there's large experimentation happening together with its pharma, academia coming together, and you share the data, you're ensuring that the experiments are done quite accurately, right? In the same way, and that is definitely a great source. So kudos to some some of these collaboration that'll come ahead. And I think to that point, right, all of, that is important. I think open sourcing, for example, I really want to give kudos also to AstraZeneca and other companies that have brought their generative models to bear and being used in many different, I would say, programs for designing new molecules. So all supportive of this, and I think it's extremely important for the scientific community to keep up data and models open source.
Yeah, one of the things I find really exciting about this field is that we've created an expectation that, well, with a few annoying exceptions like AlphaFold 3, people are going to release code along with their models. And I think it's great because we can then download this code, we can try it, but as people in industry, I think we also have a responsibility then to provide some feedback to the people who are publishing these papers and releasing their code and letting them know where it works and where it doesn't and how it could be improved. So I think there's we've got a really nice ecosystem now, and we just need to continue to build on that ecosystem.
And how do you think that feedback should happen? Like in what kind of setting, or how does it happen? I went off script, but
I mean, I think it can happen in a variety of ways. One of the things that I tend to do is just submit issues into people's GitHub repos and say, okay, I tried this doesn't work. You might want to take another look. I think the other thing that we can provide as people in industry is just a bit of a reality check on relevance, because sometimes, I'll see people, graduate students over there, who are working on problems that you kind of look and you're like, well, that's not really a relevant problem, or this data set and the way that you're splitting this data set actually introduces some bias that you may not realize. So I think a lot of it is just providing feedback that you can do either formally or informally.
Okay, last formal question is, what do you think the role of industry-academic partnerships should be in this open innovation? And maybe I'll start with you, Julie. We'll attempt to mix it up one more time.
Yeah, sure. Well, I want to play off what Pat just said, which is making sure that people with the computational skills are applying them to the right problems, right? So open innovation, where we bring together people with necessary expertise, the subject matter experts, the computational experts, the AI experts, the compute experts, and so forth, toward a common problem. So there's a so that we can make progress in a way that's really going to be meaningful. So I think that in academia, often we have a lot of the subject matter expertise around the biological problem, but maybe the pharma companies will have more information about the drug development problems and how, and then so forth. And so I think building those multidisciplinary teams can be very effective.
I think I absolutely agree on that. I think that is a fundamental thing. I think pharma companies are focused very much on drug discovery, and when you try to do discovery, I think it is too bullish to say you're also developing good models, right? I think it takes a lot of effort and true innovation to develop some of these foundational models that could be applicable. And I think this is where the academic groups really excel. And that partnership between the academia bringing in the real science, the fundamental science, which they're passionate about, and their focus to pharma companies that have data. So we sit ourselves on a lot of data, and the reason we don't try to build new models on our own is because there are a lot of them already, right, with academic groups. So we partner with them to build some of these and utilize this data that we have in driving some of the solutions. So I think there's definitely a lot of scope, and I think the time to collaborate was yesterday, but we'll do it today as well.
Yeah, so I don't have much more to add beyond what others have said. I think they're exactly on the same points I was thinking about on this topic. Yeah, I'm not going to just repeat what you said. I agree.
So I think it's important to build operational structure that can help work together. So for example, in AstraZeneca, we have multiple postdoc programs that are very successful together with the academics. I think that nowadays there's also more and more public data that could be shared in one or other ways. I think NIH could play a really important role here in mitigating that. Sorry. So I think really thinking about the operational structure that can help to do that is important. And then there's questions that are more fundamental, like when we think, you mentioned earlier, the different going beyond humans, so looking at other organisms, right? So you could think about really research programs that include how we link those different animal models or other organisms together with human, and then build programs around that. So there's many areas where we could actually collaborate, and the industry can channel those activities towards application in the drug discovery from the basic research.
Yeah, I think there are a lot of nice formal mechanisms for doing this, but those formal mechanisms often involve lots of money and lots of lawyers. And I often find it more fruitful to just get together with somebody over coffee and talk about science and talk about the problems that we're working on. So, I think that there are places for formal relationships, but for those in academia, I would also urge you to reach out informally to people in industry to just have a conversation.
Yeah, to really simplify the process, right? Yeah. Okay, so we're done with the formal questions. So now we are opening it up to the audience to ask whatever questions you would like.
Is that working? Yes. Science is not efficient. I'm particularly interested in two things. One is, essentially negative results, the database of negative results, or the database of things we tried that didn't work, and how that might improve our use of AI. The second thing is maybe less AI focused, but I think is also still important for the conversation, and is things that did work but may not be protected by IP, or maybe sitting on the shelf. I know that there's been lots of organizations that have been nonprofit that have been focused on kind of following up with good candidates that just didn't have the resources or the commercial kind of angle to be able to get them to move forward. And I'd love to hear your comments on particularly. We know there's siloization, but particularly the area of that, the hidden kind of results, which we never see because they're never really talked about, and that's the negative results, the things that were looked at but then didn't move forward for various reasons.
Maybe I'll start on this one still, since I stirred the pot. So I will say that I think we had the same conversation with somebody I remember a couple of years back, some Nature and Science publication, same question about, can we publish this data? And I think the fundamental problem is people may be willing to even publish the data. The question is, would you trust it? I think the scientific community is so, which we should fundamentally question everything that we do about ourselves and about our peers as well. The question then remains, is the negative data because the experiment didn't work, or because we missed something in the experiment while conducting it? And I think that sort of then, that kind of clouded the whole effort to try to publish some of this negative data. And that's where it kind of buried. And then we came up with this solution that why don't we form this consortia and labs where we can do the experiments and share that data, right? And if the data is done more synchronously, either with some automation involved in it, definitely, right? So we remove that inaccuracies, there's more predictability on that data, then that could be published, and that could be utilized. And that's where I think this whole open innovation forum is important. I completely agree with you, but in my opinion, the only solution to this is actually generating some of this data rather than going back to historic data, because we're going to come back to the same problem of how was it done, is it accurate, do we trust it, and so on. But I'm happy to hear the opinion of other panelists here.
Yeah, I think if you have a targeted effort, you can get to what you need sometimes. And as an example, so one of the places that you never see negative data is organic synthesis, right? Because nobody wants to publish a reaction that didn't work. However, there is a lot of that data out there. There's a lot of that data actually out there in pharmaceutical companies on non-proprietary compounds, where people are just trying to establish reaction conditions for Suzuki coupling. So my former colleague, Steve Kearns, together with Connor Coley at MIT, put together the Open Reaction Database. So now they have a database, but it's not, I don't think it's enough to actually just say, great, we've got a database, the database is here. So they ended up hiring somebody to be the advocate for this and go out and talk to pharma companies and see where the data is. So I think it requires effort, but I think if you're targeted and you can figure out what you need and who has it, you can put a mechanism in place to get a lot of this data.
I'll just add on to that. It does come to incentives, right? What incentives do people have to share this data? And I think you're right that it's going to be hard for people to passively do it. The incentive isn't there. So an active effort to get such data, I think, would be needed.
And on that incentive point, I can think of one experience I had, and I forget what institute it was that did this, but they had a nice model where basically industry partners would pay in to create a pool of money that they could use to then give out grants to fund experiments to academics. And what industry got back in exchange is we had access to that data. So there we weren't restricted with they actually found a biological story that led to a publication, and then they released the data as part of it. It didn't matter if they found a story around it. We had access to the data. So maybe that's one way to incentivize it, is that actually the data generation itself is what gets funded, and then the data that's generated is made available to the entire community, not just the people who paid in, maybe.
Yeah, I mean, sort of somewhat related to this, right, is that, in industry, you often go out and get a paper right, try to reproduce the results, and how many times is it not reproducible? So that, I guess, maybe feeds into what you were saying, right? But I don't know about trusting the negative data or not seeing the negative data. So I don't know if people have any thoughts on that.
Just want to add on the incentive also, right? So when you have negative results, then the narrative, the story you want to tell is much less attractive. So for instance, when you think about publications, right, you're probably not going to publish in Nature or Science negative results. They're not going to even look at it, right? So I think that's part of the problem. You're right, you need to stimulate some more incentives for people to be able to share this kind of data or invest in the business to share it, because that's in the end it requires a lot of investment. Now, on labels on approved drugs and stuff like that, that's something that is obviously much more complicated to share. Yeah.
Somebody told me, and I'm sure this is for all pharma companies, that if the experiments fail or the compounds don't go through the clinic, that's the only way for them to publish. I don't know if that's true, not true. You guys can disagree with me.
No, that's not true.
So that's
whole mechanism set up for publications of also things that are actually working very well. It's just the different process, but it happens a lot.
Right. So if it is, so that's all positive to hear, right? And so I think if, then again, it comes back to the point that I think the more we can, to the same question that was asked earlier as well, can we publish some of these compounds that did make it to the clinic and probably were or weren't successful in the process? Probably if the funding was lost in the process because of change of strategy. And this was just my more provocative way to say maybe pharma companies should do that a lot more than probably they're doing.
Other question?
Hello, James Kozlowski, IBM Research. I just wanted to ask, because you all are in the position of deciding which data gets collected next in your respective organizations, do you see AI, and in particular, the opportunities for uncertainty quantification, helping you in that important decision? That is, which data to collect next that will have the biggest impact on reducing uncertainty, in other words, provide the most economic value to your decision?
Absolutely. So I think this has been one of the fundamental changes in my work over the last 7 or 8 years. So, earlier in my career, people would run experiments the same way they always did. They'd give me the data and say, here, build a model. And I'd say, that's not the data that I needed. So, one of the things that we did early on was made sure that we had budget allocated for experiments to improve models. So, one of my favorite machine learning techniques is Gaussian process regression. One of the things I like about that is that you not only get a prediction, you get an uncertainty. And you can use those uncertainties to guide additional experiments. I mean, especially things like pharmacokinetics. So we typically don't put every molecule into a PK study because it's expensive. However, we can then build a model, look at the regions of chemical space where we're uncertain, and then say, okay, great, we don't really know a lot in this area. Let's take 5 compounds and put them into a cassette dosing PK study. And we've found that this can consistently improve our models. So yes, I think we really need to have a very close coupling between the people building the models and the people running the experiments.
Yeah, one of the areas I will add that we're working on is on Bayesian optimization for medchem, for catalysis especially. So we do look at, that is one, that has been one of the, so not really DOE, but using Bayesian optimization to decide which catalysts work really better with which kind of compounds that you're working on, or your building blocks. And looking at the yield, like your classic conditions, like you need better yield, you need what catalyst combinations are going to work, what aren't going to work, and especially when you're trying to scale up certain reactions on the process chem side, this is super, super, super helpful. So that is, I would say, a very specific problem where you need the models to, very specific models, and also, experiments to decide what kind of or rather the endpoints to decide what kind of experiments you want to run in that area.
So I agree with what you said. I think that that's absolutely necessary, and the AI plays an important role. I want to add uncertainty, incorporating uncertainty in the decision process of organization is a cultural change, right? I think that going back to translational medicine, right? Translational medicine in many aspects is really exploring the territory and see where you could bring, measurements that can inform the, the study and the drug, right? And benefiting obviously patients. So I think that that's something that you want to expedite with AI. And I think one of our visions in AstraZeneca is this, what we call the augmented scientist. And the idea is that we want to augment scientists with information and knowledge from different areas. Obviously, LLMs and stuff can help do that better in human-machine interaction. And I think that there you could augment a scientist with, okay, I will need that data to help me make a better decision, more informative decision about this specific area. And I think that that's something that is evolving, and AI definitely going to is playing a big role there.
Yeah, I guess the only thing I would add to that is maybe, because I agree with everything everyone already said, the flip side of it, I guess, is for the modelers to maybe move towards more models that provide those uncertainty estimates, right? There are models out there now that will just be confidently wrong and you'll never know it. And with those, you can't really apply this approach. So I think it requires activity from both sides to make that work.
Any other questions? I saw, oh, okay.
Go ahead. Yeah. So I'm hoping we can actually make this a little bit more actionable. We all agree that we need more data. So what's the next UK biobank? What's the next ADME that we can agree on? Are there 2 or 3, or are there 20 UK biobanks? Because there's these models are so data hungry. Can we agree on 2 or 3 leading ones? And can we even start to think about how we would power them? Right? How big do they need to be? What does this effort need to look like?
I can start from my side of it, which is, again, the single-cell omics perturbations, right? So I think there's been an effort, even proposals in the literature towards building a perturbation atlas. Part of what I think the models suffer from at the moment is that most of the data that's been collected, it's sort of convenience data for us because it's available already, but it's looking at a very narrow range of natural biological variation. And I think the models fail when they're confronted with something very off, like an entire gene was just completely removed, but the other ones were left alone. So efforts to systematically generate perturbational data, both genetic perturbations, chemical perturbations, and start to accumulate that in some sort of atlas would be very helpful.
I would put more investment in really generating longitudinal clinical data. So having much more accurate, higher quality data coming from real world in a way that would be ready. And then if you do that long enough and on a big enough population, then you will get enough, what we could say, perturbations from natural disease that comes along. And this will enable us to understand better the human biology, human talks, and link it to other places, right? So, I mean, keynotes mentioned earlier the food. The food space, where you could actually use that to understand better, toxicity profiles, right? Obviously, if you have more data that can help you understand that space better, that can be helpful. Again, it's hypothesis, so I don't know, but I think, yeah, longitudinal data would be very helpful.
Yeah, this is a great question and a very hard one. I mean, my mind, coming from NCI, first goes to cancer and how, the model we heard in the keynotes, that some of these models, like the single cell models, are based on quote unquote normal cells. And so, do we need to focus on particular cancer types when we think about the cancer space? I think longitudinal is a great point, but also multimodal. Can we generate data sets that across on samples that collect data through multimodes, multiple molecular imaging, organ, and so forth, that we can bring together for exploration.
So thank you, first of all, for this question. I think we all chat about things, and there's nothing tactical that comes out. So appreciate that. I will say we're doing 3 tactical things, and this is also an open invitation for collaboration. So I'm going to be here, so I'm happy to follow up on this. The first thing that we have done is we have released the models that we have trained on our own ADMET data. So this is last 30 years of ADMET data. Again, it had to be distilled down to a few ADMET endpoints because a lot of it was garbage. And as always in all pharma companies, it happens. And so we trained on very specific ADMET. We have 20 ADMET endpoints that we work on, and we trained the model on this pharma data, and we've released it into a product called Addison. The second thing that we have done tactically, again, it is going to be very focused on oncology and immuno-oncology because that's our domain, that's our therapeutic area, right? So if you try to use it for rare disease, it may not work that way, which is okay, right? Which is part of the process. You have to be realistic how your models work, but you can transfer learning to other modalities. So that's one tactical thing. Second, we are building about 20,000 square feet of automation that is purely functional. Part of it is going to be fully for ADME. So every compound that's generated, it's about 250 molecules a week, will go through that 20 ADME assays, whether they're required or not. And so imagine now this exponential growth of data coming from every compound that you generate. You have those same ADME endpoints run in the same exact way, and the only way to do it is automated. And so we will be, you will soon hear about that as well, but like I said, we're open to collaborating very early on as we are in the development phase. And then the third tactical part to this is all the data that's going to be generated from those ADME assays will go back into the models. And that's how we can expand from a particular therapeutic area, which is oncology, immuno-oncology, to other areas. And that's where we need those collaborations where other companies that are really well working on other therapeutic areas, rare disease, to partner with and use this mechanism, to generate more data. So three tactical things that I can think off the bat, and happy to collaborate on some of this.
But if that's taking place in a closed environment, it's not helping the field. If you take a look, what's really tripping us up and what's slowing us down in drug discovery? It's pharmacokinetics and tox. And we need to just generate large, consistent data sets and make them publicly available. So we need to generate data for aqueous solubility, metabolic stability, data on drug metabolizing enzymes, data on off-targets like hERG, data on cytochrome P450s. Just make this data available. I'll finish with federated learning is not the answer to this. There was a large European pharma consortium that essentially took $20 million and lit it on fire. Nothing came out of that. It's not the way. We need to just generate this data and make the data publicly available.
All right, maybe one more quick question.
Just follow the question, Fei-Chung Chen from Cleveland Clinical. Follow the question is, this panel had good discussion about the data, about the benchmark experiment assay. But the question today for drug discovery field is we have too many assays, right? And too many assays we generate, inconsistent data. So I really wanted to ask the panel suggesting how we really can work on together to generate, more benchmark or even a few list key assay or key experimental data we can feed to AI, we can help the AI, not just feeding all data we have today.
Any comments?
Right. And that's why I think I completely agree, Pat, right? I think we have to partner. And I hope after this panel, we partner. And like I said, this is an open invitation for collaboration, right? This is not a closed space that we're building. We want these partnerships and collaboration, and we're happy to make some of that data public, right, as we partner. But that's fundamentally why we're starting with only 20 admit assays and not the whole world there, because we can't do all of it, right? It's the primary assays that are common to all targets for the most part. And I agree, there are going to be a lot of other functional assays that will be run. They're going to be very specific to those targets, and that's going to take some more effort to work on. But they're also different for different targets and so on and so forth, right? So our idea was 20 assays that are fundamentally needed for everything. Let's start with that. Let's get that running and functioning, and then happy to expand that. But I agree with you. It needs to be focused.
Anyone else? Well, I think one thing you mentioned was we talk a lot about some of these things, and then there isn't necessarily follow-up. So I think today's meeting is really about the follow-up, about getting people interested, figuring out what areas we should work in, and forming this AI Alliance working group. So you'll hear more about that later, but I hope people also sign up for that. So I'd now just really like to thank the panelists for a really great and lively discussion.